So we starting with the genetics, and I just want to tell you guys that the order of terms in this lecture matches the order in first day, so it will be more convenient for you. So we will start with gene expression modifications, right? And first I want to start with explaining what gene means, right? So gene is part of the DNA, right? So when you express this part of the DNA, you make certain product eventually, right? Proteins. So when we express DNA, right, gene, we go through process called transcription, and we make RNA, and then we go through process called translation, right, and we make protein eventually, our product, right? And if you want to go deeper on this topic, you can check out my previous videos on this topic, but today we're talking about gene expression modification. So our matrix here is DNA, right? What we do with our DNA, we can knock out gene or knock in gene, or removing gene or inserting gene, right? If we insert certain gene, we can randomly insert it or targetedly insert it. When we randomly insert it, we constitutively express the gene, non-stoppably express the gene and make the product, right? When we targetedly insert the gene, we conditionally express it. So we make certain amount of product. So that's the difference. Next is RNA interference. So here our matrix is RNA, right? And we regulate process of translation here. So what we do, we work with small non-coding RNA target, which targets matrix RNA, which is coding, right, to inhibit gene expression. So we have our micro with me, right, microRNA, and small interfering, which is cRNA. What we do in case of microRNA, it is naturally produced by cells as hairpin structures, right? What it does, it binds to matrix RNA and inhibits translation. It can occur normally when it functions to facilitate degradation in our normal cells, right? And abnormal expression of it can occur when it inhibits tumor suppressor gene. And when it does it, we don't have cells that limitly divide, right? So for example, if we have pathologic cells, it will facilitate, like, express tumor suppressor gene in order to stop it from dividing and progressing. And if we block it, this process of tumor suppressor gene expression, these cells will uncontrollably divide and tumor will progress, right? Therefore, normally for degradation and pathologically for certain malignancies development. Small interfering RNA derived from exogenous double-stranded RNA, right? And it's highly specifically binds to certain sites of mRNA, which results in cleavage of those prior to translation. And it's used mostly for gene knockdown experiment, right? When we work with mRNA, bind to certain genes in order to decrease their expression, right? So it works like that. Next is genetic terms. You have to know and distinguish certain genetic terms in order to solve most genetic questions in USMLE, right? So what codominance means? Codominance is where you have like not only one gene or allele responsible for phenotype of this heterozygotes, right? Multiple alleles responsible. So for example, blood group, you have A, you have B, right? AB, you know, group types. Then alpha 1 antitrypsin is very high yield for USMLE. Alpha 1 antitrypsin is actually regulates degradation process in the lungs, right? It's synthesized in the liver and then it moves all the way to the lungs in order to function there. So if you have pathologic alpha 1 antitrypsin, it stocks in the liver and damages it, right? So you have liver dysfunction and increase degradation process in the lungs, which will cause emphysema of the lungs, and you have liver dysfunction lab findings in your stem, and you automatically assume that is alpha 1 antitrypsin deficiency. So when we talk about genetics, it's like ZZ, which is responsible for development of this disease, right? But when you have MZ, you don't necessarily have the disease. You have a susceptibility of developing of the disease. So when you get exposed to certain risk factors such as cigarettes, alcohol, you will develop in early age this disease, right? So be aware of this, and MM is actually normal for alpha 1 antitrypsin. HLA groups is compatibility, right? And we know that not only one but multiple alleles contribute to certain compatibility HLA groups. So variable expressivity is when patients with the same genotype have varying phenotypes, right? It is important to distinguish variable expressivity and incomplete penetrance because it can be very, very similar. But if question asks you about neurofibromatosis, it's always variable expressivity, even if both present in answer choices, right? It's first point, remember that one. Then variable expressivity, it's actually when everyone in the family, like in children, all children have the disease but varying presentation of the disease. For example, neurofibromatosis, lesion in your eyes. For example, one child have it, then the second one have capillary spots on their body. The third child have skin freckles, right, in the groin or in armpits, right, something like that. So we have variable expression of this disease, variable presentation of the disease in different children. This is variable expressivity. Incomplete penetrance means that one child have the disease, the other don't have any, right? It is true for example, for BRCA one gene mutation, not only BRCA, right? So breast and ovarian cancer can develop not in all children who have this gene mutation, right? This is like also Angelina Jolie had BRCA one gene mutation, so therefore she removed her breasts and ovaries, just so you know and remember. about BRCA1. So next one is pleiotropy, is when one gene, right, contributes to multiple phenotypic effects. What that means is that only one gene mutated, right, but it causes light skin in PKU, musty odor, intellectual disabilities. We have PKU, which means phenylketonuria, which presents was not only one presentation, that what that means, right? Next we have anticipation. Anticipation equals accumulation, right? So it means increased severity or earlier onset of disease in succeeding generation. It is true for trinucleotide repeat disorders, for example, Huntington disease, right? So we know Huntington is mood swings, depression, tremor, right? Then something with muscle weakness, something like that, right? Huntington. So one patient will have Huntington in symptoms in his 60s. Then his son will have it in his 50s. Then his grandson will have it in his 30s. And severity can even worsen in those succeeding generations, right? This called anticipation. So usually you will have Sam explaining you that father had the symptoms in this age and his son had the symptoms in this age earlier, right, and more severe. And then they ask what the process called anticipation. Okay. Then a loss of heterozygosity is like, for example, two-hit hypothesis in retinoblastoma is most tumor suppressor genes require both alleles, right? So retinoblastoma develops when you lose your tumor suppressor gene. So tumor suppressor gene works if it has heterozygous, right? One normal, one pathologic, or two normal genes. So if you have only one gene mutated, one hit, right? You will not remove your tumor suppressor gene, and therefore you will not develop retinoblastoma. If you, for example, born with certain mutation of one gene, one hit, and then you were exposed to certain risk factor, right? And you get your second hit, and then you developed retinoblastoma. That called two-hit hypothesis, right? Therefore, it's called two-hit hypothesis. So, in order to tumor suppressor not being function, to your genes should be hit, let's say, right? Pathologic. So both alleles to be inactivated. Should be both alleles inactivated in order for tumor suppressor genes to be function. Next, we talk about to be inactivated, right? In order for tumor suppressor gene to be inactivated. Same thing happens with mismatch repair genes. Mismatch repair genes, if we have some pathology in those, we develop Lynch syndrome, right? Or non-polyposis tumor syndrome, we call it. MSH, MLH, PMS, it's all Lynch syndrome. Then TP53 gene mutation, which produces P53, which is also tumor suppressor, right? You develop leaf from many multiple tumors in one person or generation, like not the same type of tumor, but different type of tumors, right? It is leukemia. Loss of heterozygosity is mostly because of tumor suppressor genes. Remember that one. Next is question. A 39-year-old man and his wife have two sons with history of retinoblastoma and the other with osteosarcoma. Neither the patient nor his wife have history of health problems, right? The patient is identified as 2 and 5 on the pedigree. So this is the patient, this is the father and his wife, right? They have healthy and children with the disease, right? So we look at the big picture and see that the problem in the father, right? Because he had family and run disease in his family. So we look at the answer choices here. Germline mosaicism. Germline mosaicism means that father had mutation in his sperm cells, and therefore one cell's mosaicism means one cells are normal and other are not normal. So other are underwent certain mutation, and therefore some children will be healthy and other will have the disease. It will be true for this case if his family were in if his family wasn't present with disease cases, right? So therefore it's not the case here. Heteroplasmy is when we talk about mitochondrial disease first of all, and then only mothers can pass it to their children. For example, mom here will be sick and she will pass the disease to her children, and then her son sick here, he couldn't give to anyone, and her daughter here and here or here, if she would be her daughter, she can pass it if she sick, right? Etc. etc. Incomplete penetrance. Incomplete penetrance is when our case here, for example, right? Certain children have disease phenotypically present, but certain don't have it phenotypically present. So all of these children have the disease. They all like genotypically, but phenotypically not all sick, right? So therefore, this dad have genotypically passed the disease to his children, but not all of them are sick. It is incomplete penetrance. Somatic mosaicism is when, for example, you have a child with certain features of Down syndrome, but you're not sure, but suspicious of him having Down syndrome. And then you take a swab, do genetic evaluation, right? And you see that certain cells are normal and certain cells have abnormal amount of chromosomes, right? So 21 chromosomes is mutated. This called somatic mosaicism. The difference with genetic mosaicism, germline mosaicism, is that germline mosaicism is about your gonadal cells, right? Sperm cells or ovocytes, whereas somatic mosaicism is about your body cells, right? In your body. Variable expressivity is very similar to incomplete penetrance, but as I said, it is associated with neurofibromatosis type 1, first of all, and second of all, if it's similar. It is shown that all children have certain presentation of the disease, right? In varying severity or varying presentation. Okay, next one is epistasis, not epistaxis. Epistaxis is when you're bleeding from your nose, right? Epistasis is unrelated gene that modifies the phenotypic expression of another gene. So you have to look at the example here. So for example, dominant A is contributes to brown color of your hair, right? Dominant B contributes to black color of your hair. But if you don't have dominant C, which is contributes to coloring, you will not have brown or black hair, even though A or B is present. For example, AA, BB, dominant, CC. AA dominant, you have to have brown hair, right? And if CC dominant, you will have. Then BB is dominant, you have to have black hair only if CC is dominant as well. Then AA dominant but CC is recessive, you will have white hair. Or BB is dominant but CC is recessive, you will have white hair, etc., etc., right? So unrelated gene here is CC because it do not contributes to your actual color, but it contributes to presentation of this color. So this color will be brown if CC is present only. Aneuploidy is when non-disjunction occurs of your chromosome during meiosis or mitosis, doesn't matter, right? It will result in abnormal number of chromosomes in Down syndrome, Turner, or it will contribute to tumor developing, right? Oncogenesis. Dominant negative mutation is when this occurs, heterozygous produces non-functional altered proteins, right? I will show an example here. So here we have homozygous, right? Normal product, and it will mix with product of second gene and produce normal tetramer. Then we have heterozygous with mutated product, which will mix with second gene product, and we have product that is pathologic and non-functional. It is true for P53, right? If we have only, it is tetramer, only one protein mutated, only one piece of it mutated, the whole tetramer is non-functional.